All right, everybody, welcome back. Welcome back. We are beginning the second portion of this wonderful Stargardt Summit. And thank you to everyone who is joining us virtually once again. As noted, this is the Stargardt Super Bowl, and so we're starting a little bit late because the bathroom situation was just like going to the Super Bowl there. Never had a line at a conference, but always something new, and we are just so excited to be back here with one of the more anticipated aspects, elements of this summit. A lot of folks tuning in just for this. It's time now for the research panel. This panel brings together leading experts who are at the forefront of Stargardt disease research, treatments, and emerging innovations. Today, our panelists will discuss the latest advancements, what's on the horizon, and what these developments mean for our community. Leading this important conversation is Dr. Michael, not Marcy Green, not Marcy Blue, but Marcy Yellow, who will guide us through the discussion. Please join me in welcoming our research panel.
Well, thank you, Adam. I've never been introduced as Marcy Yellow because in Stargardt's disease, of course, we lose our color vision, so I'm not sure what color that is. But I thank you for having us, and we're looking forward to bringing some information to you. I'm a practicing physiatrist. I'm a physical medicine rehabilitation specialist who works here in Massachusetts, and I'm honored to be sitting up here with three incredible clinician researchers who are dedicating their ophthalmology careers to seek treatments and cures for people with inherited retinal diseases such as Stargardt's. I'm just as interested in you as you are hearing the information that they're going to provide because I have Stargardt's disease. I actually been diagnosed about 13 years ago at age 45. A little bit atypical that we've heard of so far. So I've only been legally blind for about 3 years, and my vision is slowly declining, but fortunately have been able to take on a career in medicine because of the late onset. So we are interested to know how this is going to play out in relation to the research that we are going to hear about today. So I'd like to start off a little bit today of just having our panelists introduce themselves and give us a little bit of your background in relation to your connections to Stargardt research. Dr. Huffeld, why don't you start?
Great, thank you. My name is Rachel Huckfeld. I'm a clinician scientist at Mass Eye, so I had the shortest drive of anyone on the panel this morning. I feel so fortunate to have a job that really puts me at the interface of science and medicine. I think like others on the panel, I have a strong research, like basic science research background, and now I get to take care of patients, including some people who it's wonderful to see in the audience, but I also get to help bring this really groundbreaking science that's leading to investigational therapies to those patients. So maybe like Greg said, I think I found a sweet spot for a job.
I'm Kenneth Fan. I'm from Houston, Texas. I work at a retina private practice there, and I see all our inherited retinal disease patients there. I did my training over in Miami, where I met an amazing mentor who really showed me the importance of inherited retinal diseases and the need that we have in that area. I just want to say that, of course, I was invited here to maybe help you guys learn a little bit more, but truthfully, honestly, through the talks this morning, I think I've learned more from you guys than I might be able to give back to you. So this is a truly amazing opportunity and symbiotic relationship, and I'm looking forward to meeting you guys.
All right, I'm Brittany Scruggs. I am a vitreoretinal surgeon and researcher. I'm an inherited retinal disease specialist at Mayo Clinic in Rochester, Minnesota. And I have a 5th grade daughter named Lucia. But it was really wonderful to get the opportunity to come here. I wear a lot of hats, like many of us do. I'm a clinician, I'm a surgeon, and I'm a scientist. So I have a lab, a regenerative medicine lab at Mayo Clinic, and I study gene therapy and stem cell therapy. And like many of us, we are running clinical trials and seeing patients and trying to make the hard decisions of how do we advance science and what trial is best for patients. So I'm really glad that we get to have this discussion. We're still learning a lot about the science of gene therapy and different advances for Stargardt disease. So I think there's a lot of discussion to be had.
Thank you. So if we take it to, we have this condition called Stargardt disease, but Dr. Hartfeld, if you could just, what is Stargardt disease in perspective in relation to medicine and science?
Yeah, so I think my goal, I think anything I say, a lot of you are going to know already. So my goal is just to kind of start with a foundation that will be the basis for some of the other topics that we want to come to. And just to make everything, accessible and useful for our conversation today. And I'm gonna deviate for a second because I had wanted to remember to just thank Greg and the Carroll Center for putting this event together and for all the sponsors and and everyone who's here. What a special place to be today. So, Stargardt disease as a condition was recognized over 100 years ago, and I think those of you living with it don't need me to tell you what it's like to try to tell you how it affects vision and that type of thing. I'm going to stick more to, what is happening inside the eye and inside the retina. The gene that causes Stargardt disease, ABCA4, was discovered actually not that long ago, not more than less than 30 years. And Paul was correct. It is scientifically proven that people with misspellings in this gene have, higher levels of grit, resilience, and creativity than anyone else. That's science. The other thing that this gene does is it participates in the biochemical reactions that happen every time light strikes the eye. So genes encode proteins. The ABCA4 gene encodes a protein that sits in the antennas, so to speak, of the light-sensitive rod and cone cells. And again, every time light strikes the eye, it starts this cycle of biochemical reactions that all involve vitamin A and the recycling of vitamin A. And when the ABCA4 protein doesn't work all the way, it results in the accumulation of abnormal byproducts of that recycling pathway. And again, the antennas of the rods and cones, that impacts their health, and it eventually impacts the health of the supporting cells underneath, which are called the retinal pigment epithelium, or RPE. And what we see clinically as a manifestation of that as ophthalmologists is often little yellow deposits in the retina. They don't have to be there, but again, they're what we see visually. And that is the actual visual manifestation to us of those byproducts accumulating because of this faulty recycling pathway. And some of that pathophysiology we're going to come back to a little bit later as we talk about ways to intervene and try to treat Stargardt. I think everybody here probably has a good sense of the genetics of Stargardt, but I'm going to spend a moment on it anyhow. The ABCA4 gene is a big gene. You can think of genes as being like volumes of encyclopedias or encyclopedia sets. This one has way more than 26 volumes in it. And so there are over 500 at least different misspellings in this gene that can result in Stargardt. Because it is a recessively inherited condition, that means that it takes both copies of the gene, one from mom, one from dad, to be misspelled to result in the condition itself. Again, I think much of what I just told you will be familiar.
Thank you. Thank you. So let's say that I'm a research company and I have this condition called Stargardt disease, and I wanted to start developing a project and research to answer a question that we have. Dr. Pham, what's the process that takes place, and why does it take so long to get it going?
Yeah, traditionally, clinical trials in more common diseases can take a very long time, like over a decade, sometimes from first testing in animals all the way to approval by the FDA. Thankfully, for rare diseases, there are fast track designations or accelerated approvals, but essentially, you're talking about taking something that has potentially never been tested in a human before, putting that into a human setting, and then accelerating it all the way through maybe 100 or 200 or even more patients with a rare disease. And so you're going from phase 1 clinical trials, which is first in human, and usually in that stage, you're looking at getting the dose right and making sure it's safe for patients. Then you go from phase 1 to phase 2, where you kind of narrow down the dose a little bit and make sure the efficacy is there. And when you show efficacy in patients, you go from phase 2 to phase 3, and phase 3 is really the pivotal trial where you look for efficacy and potentially approval by the FDA. There are some groups that can sometimes skip phase 3. Alkeus is one of those groups that potentially we might hear something sooner, but I think that by and large, because you have to go through all 3 phases, it takes a very, very long time.
So we have a condition. We have Stargardt disease. We have a clinical trial hopefully underway. We need subjects. We need people. As I mentioned, I think I'm an outlier. I've been diagnosed at age 45. And we saw just incredible couple examples earlier already about this is a juvenile process that typically we think about Stargardt's as a young person's macular degeneration. Dr. Scruggs, could you go through this age variation? Is it a different disorder? Is there a different process in the medicine to think that why I may have been diagnosed at age 45, but we see others at age 7, 9, et cetera?
Yeah, it's a good question. And to answer that, we do have to go back to what Dr. Huckfeldt was talking about with genotypes and phenotypes. So to kind of explain what that means. So genotype is essentially the misspelling. When we do DNA testing, we do genetic testing, what is the misspelling that caused the disease? And with over 500 variants, I'll use the word variant and not mutation, there are various phenotypes, which means this disease presents in a lot of different ways across the community of patients who have Stargardt disease. The classic form of Stargardt disease is the juvenile form, and 2/3 of patients with Stargardt disease will present either in childhood or adolescence. Okay, which means 1/3 of patients will be either after young adulthood, even up to 60, 70. I had a recent diagnosis of 83. That patient had been misdiagnosed as age-related macular degeneration for several years. And I think many of us know that it is a mimicker, and some people can look at it and say, oh, this is just macular degeneration, it's nothing to do. But really, when you think about the genetics, it's different. It presents differently, it looks different, and there's more counseling to be done. So with that said, the genotype is really important. So out of those 500 variants, some are very mild. There are people in the world who have 2 ABCA4 variants that are so mild that they never present with Stargardt disease. And that could be up to like several % of an entire population just because that variant is so common in their community. So if someone has a mild variant, and when we talk about variant, what we're talking about is how functional is the protein, right? So the ABCA4 protein is really important. It's important for vitamin A and kind of transporting and allowing the visual cycle to complete and not having this deposit build up and cause toxicity. So with that said, if the protein is working at a really high level, then the patient's not going to progress as quickly, and they likely won't present as early. However, if the protein is completely not working, say someone has not a mild variant, but a more severe variant, partnered with another severe variant, so 2 severe, then that disease is going to be more advanced, potentially an earlier onset, potentially faster progression, potentially more atrophy in the macula. And so when we look at these clinical trials, and the reason we're talking about this is because there are many clinical trials out there, and you'll see that the criteria are very different, right? Some, it's a very small age range, 12 to 20, and then others, it's only adults. Some, the atrophy has to be super, super small, and others, the criteria different. So it's all because we're trying to have a population in that trial that is fairly similar. Potentially their genotypes are similar, so that we can study it better. I will say we're still learning a lot about that genotype-phenotype relationship, but there's absolutely a correlation. It's interesting, and I'm sorry, there's just so much to talk about with this, but if you have a very severe form of ABCA4 variant partnered with another, sometimes it doesn't even present as Stargardt disease, it presents as retinitis pigmentosa. And so there's a wide range of phenotypes here. Cone rod dystrophy, retinitis pigmentosa, and then milder forms that don't present until adulthood. That was a lot. Does that answer the question?
I think it does. I think I'll take that to heart. I went through that experience when I had more and more vision loss, going to my ophthalmologist in a second opinion, and they weren't quite sure. And I said, the information that came back was, I think you have Stargardt's, because we're not quite sure. So we finally got the genetics. Fortunately, nowadays, the genetics is huge. As an old guy like myself now with Stargardt's, the genetics were not offered when we were younger. So that's nice to know that we can get these genetics sooner and I do have the mutation in a couple genes locations that do have ABCA4 abnormalities.
Michael, I was just going to say, that seems to be a very common theme today among the attendees and a lot of the patients here. There was an article published not long ago, I think, out of the University of Iowa that found that the diagnostic voyage, the diagnostic odyssey of patients with inherited retinal diseases is 6 to 7 years before they get the diagnosis right.
So that does raise the concept. So, Dr. Huckfeld, what's out there currently? What clinical trials are being done? We often hear that if you just go, you're interested in trying to find out, what study could you get involved with? Look up clinicaltrials.gov. But what does it all mean? What's out there that we could potentially put ourselves or our children or our family members into these trials?
Sure. And I'm going to, before I answer that question, I'm just going to make 2 statements. One is that we heard the word stubborn from Ellen earlier. We are all stubborn too, right? So as you hear about the types of potential therapies that are under development, remember that those of us that get to work on them are stubborn. The second thing I'll mention is that any of the investigational therapies I get to tell you about are truly built on decades of careful, deliberate, federally funded research into just basic premises of science and molecular biology. So we have that foundation of research to thank for anything I get to tell you about. But we talked about genetic testing, right, and genetic diagnoses, and the obvious premise of precision medicine, to use that phrase, is precision therapies. And so many of us hope that we will have a genetic therapy for Stargardt disease where we are, correcting the DNA, its intermediary RNA, or the final protein to restore that ABCA4 protein function in the cell. And those of you who have been in this for a while may remember that at least a decade ago, a company called Sanofi tried to do this. They tested a gene therapy for Stargardt disease in a phase 1/2 trial. think we can say it was safe. I think for business and science and medicine reasons, we don't know much more. But it's an important kind of first step in understanding any of the genetic therapies that are being tested right now. And some of you may know that for inherited retinal disorders, we actually have one FDA-approved genetic therapy success so far. It's called Luxturna, for a totally different condition than we're talking about today, but the bones of it are relevant. And so Luxturna uses a modified virus called AAV as really a delivery truck to bring genetic material to the retina. Viruses are evolved to deliver DNA, and so now we're capitalizing on that by modifying that virus so it's not disease-causing, it's just a transport tool. So what Luxturna does is uses this AAV virus to deliver a correctly spelled copy of the relevant gene to the retina. And great proof of concept in every which way possible. The challenge with Stargardt is, again, the encyclopedia set is too big. The gene is too large to fit into the AAV virus. That's why the company Sanofi, over a decade ago, tried this using a different kind of virus. And it's not the one that we are using most commonly now in the realm of gene therapies for inherited retinal disorders. But what's happened since that first attempt is the development of all new kinds of molecular tools to get around what I think of as the big gene problem. And the approaches that I'll be telling you about, some are being brought to clinical trials by multiple companies converging on one method. Others are a little bit more unique. But maybe, I actually, the 10-year-old son of one of my patients had this idea, can't you just cut the gene in half and put it in 2 different viruses? And yes, children have the best ideas, right? And that is, in general, an approach that multiple companies are taking. You either ask the 2 halves of DNA to find each other inside of the cell, or they go about their merry way, they make their half proteins, and you ask those 2 half proteins to find each other inside of the cell. So in the coming months and perhaps year, we will see a few companies bringing this type of approach to phase 1/2, first in human clinical trials. As I've alluded to, DNA is really the starting point. I don't really care where we start as long as we get a functional ABCA4 protein. So there are also companies, again, going to that intermediary, the RNA, that is one step downstream of the DNA. So for example, can we, using AAV, deliver the tools to the cell using the same virus, deliver not the gene, but the tools, so that RNA, every single time it's made, can be spell checked and corrected to, again, make a normal protein. And that approach is in a first-in-human trial. Another way to target RNA is with little pieces of synthetic DNA that impact how it is read out to protein. Those are called antisense oligonucleotides, and that is an approach we've seen for other inherited retinal disorders, and there are some wonderful academic labs trying to bring that kind of therapy to clinical trials. And then as you hear me say the word editing, you might think about genome editing and this phrase CRISPR-Cas, if that is a familiar phrase to you. That is the idea of now just bringing your eraser and red pen to the DNA itself. And so correcting that DNA, correcting the misspelling, correcting the variant so that it is correct for the rest of a person's lifetime. That is a wonderful tool in some scenarios. It may have relevance for Stargardt disease, but it is so specific to the individual misspelling that when we think about 500 variants that we want solutions for, that type of specific genome editing approach is only going to address a small number of them. So it's not out of the question, it's just not 2 trials yet. So that's the precision medicine, genetic piece of things, but that's not the whole story. If we go one step downstream of the genetic variants and their effect, can we start thinking about the pathways in the retina that are perturbed when there's an ABCA4 protein that doesn't work as well? That's where deuterated vitamin A and alkiosis therapy comes in. Can we block the process that leads to these aggregates of abnormal materials in the retina and impact its health? There have also been multiple companies over time that have tried just turning down the volume on the vision cycle, this cascade that happens every single time light strikes the eye, to see if by turning it down, we can just slow down the types of damage that happen to the retina. So again, to echo Ellen, we're stubborn, right? If it didn't work the first time, can we tweak something to make it more likely to be successful, more likely to be efficacious? And then finally, anyone who has diabetes may have at some point taken a drug called metformin. Metformin, FDA approved for diabetes, taken by many people for many different indications, actually may be beneficial for the health of the photoreceptors and that structural support layer, the retinal pigment epithelium I talked about. So our colleagues at the National Eye Institute, a branch of the NIH, have been conducting a trial to see if metformin changes the rate of change. The rate of progression. So again, two big umbrellas, right? Genetic therapies, and then one step down, oral therapies. And what I'll mention just briefly because it's a little earlier days, is the idea of vision restoring therapies. Can we replace what's been lost? Can we use stem cells, for example, to, like Dr. Scruggs works on, can we use stem cells to put back light-sensitive rods and cones in the retina? Or can we just go one step further in visual pathways and use something called optogenetics to make usually light-insensitive parts of the retina light-sensitive? A lot of information. I'll stop there.
It's so easy to understand, isn't it? It is no question a lot of details, but if we want to get back to just how do we become a subject into a clinical trial, and we think about, well, we got these smart people, got the scientists working at the bench, we got a lot of this technology being developed, but what if we read in the clinicaltrial.gov and says, well, you can be barring the criteria that you might be approved for, you have to do surgery for an eye, you have to be patched for a while, you have to take this drug for 6 months. Dr. Fan, you've been part of drug development. You've been involved with this type of research. Are there differences relative to the way in which this treatment option gets to the eye? Are there differences in relation to taking a drug versus doing a surgical intervention or perhaps an injection? And how do you talk to your patients to play that down or make them understand really what they're getting into before entering a trial?
Sure. I have this conversation multiple times every visit. I think it's important to understand that clinical trials are highly regulated. I get a lot of patients who come to me and say, I'm here for the clinical trial. And you have to kind of back up a little bit and say, first we have to confirm your diagnosis and we have to tell you more about the trial. You still have to screen and get into the trial. And one of the things that I discuss with all my patients with inherited retinal diseases is what that kind of intervention looks like. And I kind of break it up into 3 main categories. So there's surgery, and then there's clinic-based intervention, like an injection in the eye, which sounds horrifying and scary, but it's actually not so bad. We do it all the time for other conditions. And then there's more systemic approaches, like typically you hear of like subcutaneous injections, like insulin and stuff like that. But oral therapies are on the horizon as well. And I also say, in these 3 boxes, with surgery, you have the opportunity of delivering a medicine directly into the retinal space where all of the action is happening. And that sounds amazing, right? You can maybe, once we figure out how to put that encyclopedia all into one delivery mechanism, we can put that whole thing into the area where the retina is having the most trouble, like in the macula. But of course, now you're talking about surgery, talking about patching the eye, out of work for at least a week. It's a huge risk, right, that you have to be willing to take. So as with everything in life, there's risk and there's reward. And we're still trying to discover what is the best approach, and is surgery one of those approaches? Intravitreal injections are another approach, which have also their upsides and downsides. You don't have to go to surgery. They don't have to put you to sleep. You don't get all the other side effects of surgery, and you can do it right in the clinic. But then, of course, the medicine doesn't get delivered directly to the space where you have a lot of the disease action. And sometimes that medicine can disperse into the eye and cause a lot of inflammation. We know that injections in the eye typically have a little more inflammation and a side effect profile that we have to be very wary about, and making sure that in that phase 1-2 clinical trial, we know exactly what the safety profile looks like. And potentially what I would say, what most of my patients are really excited about are like eyeball related treatments, ones that you can take by mouth as maybe an introduction or just to learn about a therapy that might be able to slow or halt the progression of Stargardt. And as I was saying before, there are some exciting therapies on the horizon for that. And of course, when you take something by mouth, it affects your whole body, so safety is also still an issue there. But sometimes, the thought of actually having surgery on your eye or a needle around your eye is very scary. So I usually tell my patients about all three boxes. Of course, nothing's been FDA approved yet, but the opportunity to participate in clinical trials is very, very real, and it's pretty much here.
A quick follow-up to that point, just to tell the audience, is it not true, though, if you're thinking about a surgical intervention or perhaps, say, an injection, that one eye would perhaps be treated? You're not typically going to go after both eyes, or does that vary?
Yeah, it actually varies, but I would say most, maybe upwards of 95% of clinical trials in the inherited retinal disease space is just one eye, and they typically approach your worst eye. And it's also important to note that once that eye has had surgery for a clinical trial, it won't likely be eligible for any further clinical trials ever.
Ever's a long time. So we're hearing about this wonderful technology and aspects of new things that are going to come out for us in the future to try to help preserve our vision or perhaps restore it. But Dr. Scruggs, is there anything that we can think about? What can we do now? What can we do for ourselves just to help prolong this process? Because if these clinical phases have not really reached their safety component, or they're just starting to enter, it's scary, I'm sure, for a lot of families and patients to enter these trials, and until something comes out proven. I'm a physiatrist, so again, I'm very interested in diet and exercise. I'm a big advocate of making sure that we get people to eat correctly, sleep well, do lifestyle things. Is there research or science that will help us to say, just do these simple things to start, and as the science evolves, maybe we'll get you involved a bit later. What's the science tell us?
Yeah, I think it's a great question. And what Dr. Fan said, listening to you and your community and what was already set up here in the pairs discussion is really powerful. That it's a common theme that patients got a diagnosis and potentially left without any counseling. That is a huge disservice to the patient. And we know a lot, and we have a lot of information to share when it comes to counseling. So I would love to kind of explain what I talk to with patients. And I hope that everyone gets the opportunity to kind of sit down with your own doctor and have these hard discussions. But number one is vitamin A, right? We know that vitamin A transport is the problem. And when vitamin A can't get out of the photoreceptor and binds to another vitamin A, it leads to this byproduct production and deposit that leads to cell death. So cellular dysfunction is all related to vitamin A transport issue. So we eat vitamin A all the time, whether or not you eat meat or not. It comes in various forms, but vitamin A is very high in meats, it's seafood and poultry. And then for vegetarians like myself, we get it through beta-carotene rich foods. That's gonna be lots of green and yellow and orange foods. And so we're all getting vitamin A in our diet. And so if you just say decrease your vitamin A, that's like saying stop eating a healthy diet. So that's not appropriate either. So this is actually a part of science that we still need to figure out. I will say, I think we probably all have this experience of anecdotally, patients will come and say, you know, I've really been progressing over the last few years, and I switched 3 or 4 years ago, right when I started having decline, and I switched to a really, really healthy diet because they were never counseled about vitamin A potentially, and so their diet is really, really high in vitamin A, and that potentially correlated to their decline in vision. So I actually have a dietitian I work with. She's an inherited retinal disease dietitian, and she's amazing. And what we do is check vitamin A levels on patients with Stargardt disease. And if they're very, very high, you know, high normal or more, then that requires curation of the diet. And there are some studies that show that vitamin A rich diets, very high in vitamin A, can potentially lead to lipofuscin deposit and production over time. And this has been proven in animal models, and we're starting to understand this in humans as well. So it's tricky because healthy foods are important. Healthy foods have other good things in them, like antioxidants, right? You have lutein and zeaxanthin, which are really, really good for making sure your cells are healthy. So the number one thing is just to be mindful, to be aware that potentially if you're taking a side dish that is really, really high in vitamin A, you're getting a salmon, you're getting, you know, lots of carrots and, you know, salad, et cetera, that maybe you don't pair that with something else that's really high in vitamin A. If your vitamin A level, serum vitamin A level is low normal, probably the diet is just fine and don't really have to worry about it too much, which is why I think a baseline level is actually really important. It kind of gives that reassurance that we're doing okay, your diet is fine. In addition to diet, which I think is something that we really need to prospectively study, right? This needs to be a study that we look over, you know, 10, 20-year period and actually study strict diet versus not. But in addition to that, UV light. So there's a mouse model, a knockout ABCA4 mouse model, and those mice are, you know, the correlate to the human disease. And those mice do not develop deposits in the back of the retina if they are dark reared, which means if they are in complete darkness for their entire lifetime. That's really important to remember, because the more light that gets into the eye, the more the visual cycle is working, and the more you have that dimer, that vitamin A, coming together and depositing and causing cellular dysfunction and potentially cellular death. So limiting the UV light exposure with sunglasses, protecting your eyes. I'm actually very mindful as an ophthalmologist to limit the photography we do in clinic. My patients, I try not to do angiography and lots of things that require bright lights, at least not serially, because if it's not going to change my management, why would I put those patients through potentially something that would be toxic to them? And then in addition, just oxidative stress is important to remember. So these cells in the back of the eye are very sensitive to cellular dysfunction. So if they're not working well, because maybe their protein level, their ABCA4 protein level is low, the cells are already struggling, and they're potentially on the verge of, am I surviving or am I dying? And if you put oxidative stress, extra inflammation in their environment, they potentially could succumb to the disease. Things that are very easy to understand, smoking, right? We know that smoking is very much an oxidative stressor, and we know that patients with Stargardt disease, macular degeneration, et cetera, they advance at a faster rate when they are smokers. The opposite of smoking is very healthy lifestyle, right? Someone who doesn't smoke, someone who exercises, someone who is really mindful of their oxidative stress level. which is why antioxidants are being tried in the inherited retinal disease space, not just Stargardt disease. We're doing a trial on NAC in retinitis pigmentosa. It's for a similar reason. Antioxidants on a cellular level are decreasing inflammation, which potentially could help the cells that are in the situation of struggling and they're trying to survive. So those are most of the things. So UV light, vitamin A, diet, smoking, exercise, healthy diet, yeah. And I think going back to what Dr. Huckfeldt said, there are lots of clinical trials right now trying to help with that cellular dysfunction, including metformin, amixistat, et cetera.
In regards to nutrition, perhaps anyone in the panel perhaps could chime in. We think about, again, we just mentioned a healthy diet or nutrition. There are many people who don't like their vegetables, but we're saying we got to be cautious about eating our vegetables. But for those of us who encourage people to eat healthy and don't quite get that nutrition, is there any science about, like you mentioned NAC, which is N-acetylcysteine as a supplement? Should we recommend supplements as an alternative to get some of these other chemicals in that are antioxidants if they're not getting it in by their natural food intake?
So I'm gonna speak to NAC specifically, and some of this is style as much as black and white, right or wrong. I'm very cautious about advising people to take things that don't yet have a, robustly proven benefit for the retina itself. So I'm thinking about the conversations I have with RP patients about NAC. And also just remembering that supplements are not regulated the same way drugs are. And so, its purity, its concentration, that is not being carefully checked the way it is, for example, for like a bottle of Tylenol.
Very well appreciated, because I think about that in my patients as well, is that we always think about food as medicine, and perhaps if we can really just make sure people eat more healthy, and rather than taking the supplements, I think we do better. You had mentioned, Dr. Scruggs, about oxidative stress, but if we change the concept a little bit of stress, again, open for the panel, how about mental stress? How much is the anguish of people who have difficulties, poor sleep habits, and other things that aren't in great lifestyle medicine aspects? We do know that chronic diseases do better when mental stress is also reduced because of various reasons of how they take on life. Is there any science of knowledge at this point in relation to the mental health or the mental stresses that we all endure that may or may not affect our vision?
So mental health and in general quality of life has been studied extensively in inherited retinal diseases. We know that's true. Many studies have proven that when you combine depression and anxiety, it's roughly 5 to 6 times the general population. That's a really big deal. And then also the caretakers. There's at least 4 to 5-fold higher than the general population. And we've already heard from family members that it's led to things like panic attacks and the need for like extensive therapy. And that's really important to remember as a community, because depression and anxiety decreases quality of life. And so, we talked about exercise, right? There's actually been many studies on exercise in macular degeneration, and not specifically for Stargardt disease, but for macular degeneration in general. You can imagine there's little mice on a wheel running around. It's a cute visual image. But they actually studied rodents and macular degeneration in the lab. What we do sometimes is laser the macula to make them atrophic, right? To have little atrophic spots, and then kind of follow what happens to the atrophic lesion. Does it expand? Does it develop blood vessels, et cetera? And exercise is an extremely powerful way of decreasing the complications and the expansion of those lesions. So that's a pretty powerful study, not Stargardt specific, but macular degeneration. And we know that exercise is a powerful tool for depression and anxiety. So it's kind of all related.
Can I come back to just mental health in general? Even with inherent grit and resilience, even with the support of family and friends, I think it is sometimes so helpful for people with any kind of chronic condition to have a third party outside of that kind of inner sphere. So for example, at Mass Eye and Ear, we are so fortunate to have a psychologist at our hospital who, 3 days a week talks to people whose lives are affected by their vision conditions. And he is such an important part of what I think of as our toolkit of just helping people, live their lives fully and richly with these kinds of conditions, and just kind of buffing up the strength and resilience they already bring to it.
Great information. I think, again, as I think about my patients, and healthy lifestyles do make a huge influence upon their entire life and their family of caregivers. So I'll throw this out as kind of our last concept. Maybe each of our panel members could give their take and thought in the future. If we think about in relation to the science that's coming down the road, it's certainly very hopeful for all of us to restore vision, preserve our vision. But what's your speculation? What is the current way research is going currently to think that are we more successful, likely to have this restorative aspect? Should we have false hopes for our patients? So we really should focus on preserving what we have and get people in earlier, get the genetics potentially done earlier. So if we can stop it in its tracks, perhaps maybe that would be a better choice. What's your thoughts, each of you potentially think, how should we really focus our research and our clinical research trials? Preservation, restoration, combination of the two. What's your thoughts?
I'm greedy for all of it. I think we need to pursue all of it. Thanks. Because everybody needs an intervention. I think many of the trials right now, the goal is really stopping or slowing progression. And that would be huge. That would be a real success. But if we have a 3-year-old with a new diagnosis, let's see what we can do for that 3-year-old. If we have people who have already had significant impairment of their vision, let's see what we can do to expand upon what they have. And I'm thinking about, these awful stories that we heard from the family panel that we can all relate to with referrals we get of, this comment to parents, your kid is making it up, right? We as a field need to put that into our history and recognize these cases earlier, pediatric cases earlier, get genetic diagnoses earlier as part of our way of making sure everybody can get a treatment.
I think one of the lowest hanging fruits that we have in this field is education and awareness and exposing the world to this condition. That's why organizations like this and events like this are so important, because people don't even know really what Stargardt is. And as we learn more about it, we can get earlier and earlier diagnosis, and preventing something is much easier than restoring something, at least as it pertains to the retina. So, getting super early diagnosis from referrals from educated community doctors who can send patients to specialists like Dr. Hupfeld, myself, and Dr. Scruggs, and being able to give them, hopefully, on the horizon, some sort of treatment that can halt their progression and preserve vision for the rest of their life, that would be utterly amazing. So, I just want to encourage everybody here to share your story and to continue to educate and tell everybody, because the more we know about this condition as a community, the faster we'll advance together.
So I just want to share a little bit about Luxturna. Luxturna is not applicable to Stargardt, except for it's opened the doors for gene therapy. It really has. So it was the first FDA-approved gene therapy, not just for the eye, but for all of medicine. That's huge, okay? I'm a surgeon who administers Luxturna, And I will say if I did nothing else except for just those patients and restoring vision for those patients, if that's why I went to medical school, it was worth it. And I say that because it's such a powerful experience as a physician to be able to give something that has been shown to be safe and effective, and then to give it to potentially a child and to protect their retina from potentially having this, this progressive disease. So early, I mean, if I had to choose, I mean, early is definitely, it's great. Let's intervene before the patient starts to develop, large areas of atrophy. I share that story because it is so remarkable to be able to give a medication. But we've discussed optogenetics a little bit. So optogenetics, to date, only 11 Stargardt patients have been treated with optogenetics. And these are patients who have light perception vision or 2400 vision in both eyes. So very advanced disease, and some of them are getting functional outcomes. We're still studying that. The phase 3 will start later this year. But we need all of the science from early to late to understand. But from a physician standpoint, I am extremely excited, and I'm extremely honored to be able to provide medicine to potentially advance science and give vision back to patients. So yeah, there's a lot of hope here, and I think we're all really excited about some of the trials that are out there. So we're happy to explain and talk to all of you later in the breakout sessions.
Just want to thank you all again for providing information that's inspiring both to me in the medical field, but yet when we all, again, make ourselves, as has been discussed today, that we are a family. We're connected for ourselves, who are connected for having Stargardts, and as you mentioned, giving a medication. I feel if I can just give back and give information, awareness, we should do always to bring that awareness as you've described. Round of applause for our research panel.